Introduction. In this lecture, we will explore the complex and intricate processes involved in methionine metabolism. Methionine, an essential amino acid, plays a crucial role in various biochemical pathways and cellular functions. We will delve into the biosynthesis and regulation of methionine, its salvage and recycling pathways, and its significant role in various metabolic processes. Additionally, we will discuss the regulation of S-adenosylmethionine, SAM, the role of SAM as a methyl donor, the methionine cycle, and its importance in homocysteine metabolism. Furthermore, we will examine the relationship between methionine metabolism and 1-carbon metabolism, the impact of folate and vitamin B12, methionine restriction, and its therapeutic implications. So let us embark on this journey through the fascinating world of methionine metabolism. Methionine biosynthesis is a complex process that requires several enzymes and metabolic intermediates. It begins with the condensation of homoserine and cysteine, catalyzed by the enzyme homocysteine synthase. This reaction represents a committed step towards methionine synthesis. The activity of homocysteine synthase is tightly regulated by various factors, including feedback inhibition by methionine and adenosylmethionine. Additionally, the expression and activity of enzymes involved in methionine biosynthesis are regulated at the transcriptional and post-translational levels. Transcription factors and signaling pathways play important roles in modulating the synthesis of these enzymes in response to cellular demands and environmental cues. In addition to de novo synthesis, cells possess pathways for the salvage and recycling of methionine. These pathways are crucial for maintaining methionine levels under conditions of limited availability or increased demand. Methionine salvage involves the conversion of various intermediates back into methionine using specific enzymes, such as methionine adenosyltransferase. Furthermore, cells possess the ability to metabolize S-adenosylmethionine, SAM, into methylthioadenosine, MTA, through the action of enzymes like methylthioadenosine phosphorylase. MTA can then be salvaged and recycled back into methionine through a series of enzymatic reactions. The transulfuration pathway is an alternative route for methionine metabolism. This pathway primarily occurs in the liver and involves the conversion of homocysteine into cysteine. This reaction is facilitated by the enzyme cystathionine beta synthase and cystathionine gamma lyase. The transulfuration pathway is important for sulfur amino acid metabolism, as it provides a route for the synthesis of cysteine, a conditionally essential amino acid. Methionine adenosyltransferase, MAT, is a key enzyme in methionine metabolism. It catalyzes the formation of S-adenosylmethionine, SAM, which serves as the principal methyl group donor in numerous cellular methylation reactions. SAM is involved in DNA and histone methylation, which play critical roles in the regulation of gene expression and epigenetic modifications. The activity of MAT is finely regulated by various mechanisms, including substrate availability, allosteric modulation, and post-translational modifications. Dysregulation of MAT activity can have profound consequences on cellular methylation processes and overall metabolic health. The intracellular levels of SAM are tightly regulated to meet the demands of methyl group donation while maintaining the delicate balance required for cellular homeostasis. Several mechanisms govern SAM levels, including feedback inhibition, substrate availability, and post-translational modifications of key enzymes involved in SAM synthesis and utilization. Additionally, the availability of cofactors such as folate and vitamin B12 affects SAM levels by influencing the recycling of homocysteine and the remethylation of homocysteine back to methionine. These regulatory mechanisms ensure the precise control of SAM levels, thus maintaining cellular functions. SAM serves as the primary methyl group donor in DNA and histone methylation reactions. DNA methylation plays a critical role in gene regulation, epigenetic inheritance, and genome stability. Histone methylation, on the other hand, influences chromatin structure and gene expression by altering the accessibility of DNA to transcriptional machinery. The pattern and extent of DNA and histone methylation are tightly regulated and can have profound effects on cellular processes, including development, differentiation, and disease states. Dysregulation of methylation processes can contribute to various pathologies, including cancer, neurodegenerative disorders, and metabolic diseases. The methionine cycle represents an interconnected network of reactions involving methionine, homocysteine, and various methyl group donors. This cycle is vital for the metabolism of homocysteine, a highly reactive and potentially toxic amino acid. Through remethylation and transulfuration reactions, the methionine cycle ensures the recycling of homocysteine back to methionine or its conversion into cysteine. The proper functioning of the methionine cycle is crucial for maintaining cellular methylation potential and preventing the accumulation of homocysteine, which is associated with increased cardiovascular risk, vascular dysfunction, and other pathologies. Dysregulation of the methionine cycle can lead to a range of metabolic disorders. Methionine metabolism is closely intertwined with 1-carbon metabolism, a network of biochemical reactions involved in the transfer and utilization of methyl groups. 1-carbon metabolism utilizes various coenzymes, such as folate and vitamin B12, to carry out methylation reactions and nucleotide synthesis. Methionine serves as a methyl group donor for 1-carbon metabolism, while 1-carbon metabolism supplies the necessary methyl groups for methionine synthesis and cellular methylation reactions. The tight coupling between these pathways ensures the efficient utilization of methyl groups and the coordination of cellular processes. Folate and vitamin B12 are essential cofactors for proper methionine metabolism and overall cellular function. Folate is required for the remethylation of homocysteine to methionine, whereas vitamin B12 is involved in the conversion of homocysteine to methionine through the methionine synthase enzyme. Deficiencies in folate or vitamin B12 can impair methionine metabolism, leading to elevated homocysteine levels and disturbances in cellular methylation processes. Such deficiencies are associated with an increased risk of cardiovascular disease, neurological disorders, and developmental abnormalities. Emerging evidence suggests that methionine restriction, the reduction of dietary methionine intake, can have profound effects on health and lifespan. Methionine restriction alters cellular metabolism, the activity of signaling pathways, and the expression of genes involved in aging and age-related diseases. This topic is of particular interest due to the potential therapeutic implications for conditions such as cancer, metabolic disorders, and neurodegenerative diseases. The underlying mechanisms and the effects of methionine restriction in different tissues and disease contexts are currently being investigated. The liver is a central organ in methionine metabolism and plays a critical role in maintaining its homeostasis. Hepatocytes carry out various enzymatic reactions, including methionine biosynthesis, the transulfuration pathway, and the conversion of homocysteine into methionine. Furthermore, the liver is involved in the regulation of SAM levels, DNA and histone methylation, and the interplay between methionine metabolism and 1-carbon metabolism. Dysregulation of methionine metabolism in the liver can have profound consequences on liver function, lipid metabolism, and the development of liver diseases. Inborn errors of methionine metabolism are rare genetic disorders that disrupt the normal functioning of enzymes involved in methionine metabolism. These disorders can lead to the accumulation of toxic metabolites, such as homocysteine or methylthioadenosine, and disrupt cellular methylation processes. Inborn errors of methionine metabolism are associated with a range of clinical manifestations, including developmental delays, intellectual disability, liver dysfunction, and neurological abnormalities. Early diagnosis and intervention are crucial to managing these disorders and improving patient outcomes. In conclusion, methionine metabolism is a highly regulated and interconnected network of biochemical pathways with significant implications for cellular function and overall health. Understanding the biosynthesis, salvage, and recycling pathways of methionine, as well as the regulation of SAM and its role as a methyl donor, is essential for comprehending the complex mechanisms underlying cellular processes. Moreover, the relationship between methionine metabolism and 1-carbon metabolism, the impact of folate and vitamin B12, methionine restriction, and the intricacies of methionine metabolism in specific tissues and disease contexts broaden our perspective on the therapeutic potential and clinical significance of these pathways. As we continue to explore the fascinating world of biochemistry, it is clear that methionine metabolism holds immense promise for unraveling the complexities of human health and disease. Thank you for your attention.